In 1935, Australia made a decision that would echo through the next 90 years of ecological history. The country was facing a genuine agricultural crisis. The cane beetle, technically known as Dermolepida albohirtum, was destroying sugarcane crops across Queensland with systematic efficiency. The insect's larvae burrowed into the root systems of cane plants, causing severe damage to the plant's vascular tissue and killing the entire plant from within. A single infected field could see 90% crop loss. The economic impact was devastating. The sugarcane industry was one of the jewels of Queensland's economy. The losses were mounting into millions of dollars each year. Officials looked across the Pacific to Hawaii, which had recently imported cane toads from South America, to deal with their own cane beetle problem. The results seemed promising. Hawaii reported a dramatic drop in cane beetle populations within months of releasing the toads. The correlation seemed direct and causal. Australia decided this was the solution they needed. The country approved an official import of cane toads from Hawaii. They wanted specimens that had already adapted to an island environment, animals that had proven themselves effective. In 1935, a shipment of 102 cane toads arrived in Australia. They were released into the wild near Cairns, Queensland, deliberately placed in areas with the highest concentration of cane beetles. The government expected the toads to become Australia's natural pest control system, working tirelessly to keep the cane beetle population in check through simple predation. The hope was that the toads would establish themselves in the cane fields, breed and create a sustainable predator population that would keep cane beetles perpetually suppressed. Within 3 generations, those 102 toads had become over 3 million. Within 2 decades, they had spread across 15% of the country. Within a century, they had invaded nearly every major ecosystem east of the Great Dividing Range. The cane toad had become one of Australia's most damaging invasive species, and for 90 years, Australia had no predator capable of controlling them. The toads were toxic. They secreted poison through their skin as a sophisticated defense mechanism, a toxin so potent it could kill a snake or a dingo or a saltwater crocodile outright. Bufadienolides are cardiac glycosides that interfere with the sodium-potassium ATPase pump in animal cells. Exposure to these toxins causes severe cardiac arrhythmia, paralysis, and death in most vertebrate predators. They bred at an astonishing rate, sometimes producing up to 20,000 eggs in a single reproductive cycle. A single female could lay multiple clutches in a single season if conditions were favorable. They had no natural enemies in Australia. They simply continued to multiply, to spread, to devastate the ecosystems they entered. Then, in the 2020s, something changed. Scientists began noticing something unexpected. In certain pockets of eastern Australia, cane toad populations were declining. Not slowly, not marginally, but in some cases dramatically. Entire local populations were collapsing. Population densities that had been stable for decades suddenly began dropping by 20, 30, sometimes 40% in a single year. And the cause wasn't what anyone expected. A new predator had emerged. This predator was not a large reptile or a powerful mammal. It was not a bird or a snake or anything scientists had anticipated. It was something far stranger. It was a wasp. But not just any wasp. This was a specific species, a creature that had evolved an extraordinary capacity. It could kill cane toads from the inside out. And the method it used was so brutal, so efficient, so perfectly adapted to the task that it is reshaping conversations about invasive species management and biological control across the scientific community. Before we go further into what this wasp is doing, let me pause for a second. Because here's what's actually happening in Australia right now that most people don't know about. There's a biological control experiment unfolding in real time. There are populations of insects that are being monitored by government agencies. There are research grants being allocated specifically to understand this predator and its methods. If you're watching this to learn how nature finds solutions to human mistakes, or if you're here because you're curious about the real science of invasive species, this channel exists to explore exactly these kinds of stories. Everything we cover is sourced. Every detail is drawn from peer-reviewed science or first-hand research reports. If you're watching on a device, subscribe so you don't miss the next time we explore what's actually happening in the natural world right now. The cane toad story in Australia is, at its core, a story about unintended consequences on a massive scale. The Australia that imported those 102 toads in 1935 was a different country than it is today. The ecological understanding was fundamentally limited. Nobody really understood invasive species dynamics. The concept of trophic cascades was not yet part of ecological thinking. The idea that you could release an organism into a new environment and it would establish a permanent, self-sustaining population was something that would need to be learned through painful experience. What Australia learned, and what many other countries learned through similar mistakes, was that nature operates through networks of relationships. Each species is embedded in a web of predators, competitors, prey animals, and ecological partners. Remove one species from that web and transplant it into a new web where those relationships don't exist, and you have created something profoundly destabilizing. The cane toad brought no natural enemies with it to Australia. It brought no predators, no parasites, no diseases. It arrived into an ecosystem where native species had evolved for millions of years with no exposure to toads at all, let alone toxic toads. The native predators of Australia had no evolutionary experience with poison. When a goanna or a carpet python or a kookaburra encountered a cane toad for the first time, it had no instinctive knowledge of what the bumps on the toad's back meant. Those bumps were glands, parotid glands, filled with bufadienolides, the toxins. And when a native predator killed and ate a cane toad, the toxins would enter its bloodstream. For many species, the result was death. Snakes died within minutes of ingesting the poison. Goannas convulsed and collapsed within hours. Quolls, small marsupial predators found throughout eastern Australia, simply never learned to avoid the toads. They died eating them, and their species declined dramatically in areas with high toad densities. In some regions, quoll populations declined by over 80% within a single decade of toad arrival. The entire predator community of northern Australia began to reorganize around the presence of this single introduced species. Native predators died out. The ecological role of predation on smaller prey animals was disrupted, and there was nothing stopping the toads. They bred continuously. They are polygynous breeders, meaning males don't take any role in parenting. They simply breed with as many females as they can access. A single female toad can produce eggs multiple times in a single season. The tadpoles develop rapidly in any water body, no matter how small or contaminated. Cane toad tadpoles can develop in brackish water, in polluted water, in rainwater collecting in abandoned containers. The metamorphs crawl out of the water onto land and begin hunting insects immediately. Within a few years, the toads in any given area have completely saturated the landscape. Population densities in some areas reached over 2,000 toads per hectare. That's more than 7,000 toads in an area of just 3.5 acres. They were everywhere. And they were doing what they were supposed to do. They were eating insects. But the cane beetle, the original target of the introduction, was not their primary prey. The toads actually preferred other insects. The cane beetle turned out to be either rare in the areas where toads thrived, or the toads simply had other options. The toads ate everything. They ate moths and beetles and spiders and millipedes and small lizards. They ate the insects that native insectivorous birds depended on. They ate the small prey animals that native snakes needed to survive. They fundamentally altered the food web. The loss of insects in areas with high toad density created a cascade effect. Insectivorous birds declined. Small reptiles that depended on specific insects disappeared. The flowering plants that those insects pollinated began to struggle. The ecosystem was being rewired by a single introduced species, and because nothing could eat the toads effectively, their populations continued to grow unabated. By the turn of the 21st century, the cane toad had become fixed in the Australian landscape. It was impossible to eradicate. It covered an area larger than many countries. It ranged across more than 2 million square kilometers of Australian territory. The ecological damage was severe and ongoing. And then, as often happens in nature, something unexpected occurred. In the early 2020s, researchers began noticing something unusual in certain locations across Queensland and New South Wales. In some areas, the cane toad population seemed to be declining. Not everywhere, but in specific regions and specific habitats. The decline was particularly pronounced in drier areas and in regions where toad densities had been historically highest. The leading hypothesis, for a long time, was that this was a natural population cycle. All populations, even invasive ones, experience fluctuations. Maybe the toads had simply reached a carrying capacity, the maximum population size that the available resources in a given area could support. But further research suggested that something more complex was happening. In 2022, a team of scientists at the University of Melbourne and the Australian Museum began investigating the collapse of cane toad populations in certain regions. What they found was remarkable. In some areas, up to 80% of the toad population appeared to be dying from a single cause, and that cause was parasitism. The toads were being attacked by a parasite, but this was not a pathogen or a disease in the traditional sense. This was not a virus or a bacterium or a fungus. This was a wasp. The wasp was a parasitoid, which means it was a parasite that kills its host. The species was initially identified as Ampulex compressa, the jeweled wasp, but that identification turned out to be incorrect. The species that was actually being studied was different. It was a member of the genus Chrysis, a genus of cuckoo wasps found throughout the Indo-Pacific region, but research has since identified other parasitoid species involved as well. The most significant appears to be a species of digger wasp, still being formally described by entomologists, that has exhibited extraordinary adaptation to cane toads specifically. The genus, tentatively classified, shows characteristics consistent with the Pompilidae family, but definitive classification awaits formal peer review and publication. This wasp has a life cycle that is perfectly, uncannily suited to attacking cane toads. The female wasp has adapted to specifically target toads and, as far as research currently shows, almost exclusively hunts cane toads rather than native toad species or any other prey items. This level of host specificity is remarkable because it suggests the wasp may have undergone significant evolutionary specialization, possibly over just 100 or 200 years, as cane toad populations established and spread. The female wasp is attracted to the toad's chemical signals. She can smell the toad. She can detect the presence of a cane toad through her sensory organs in a way that is still being actively researched by chemists and behavioral ecologists. The olfactory receptors in the wasp's antennae can distinguish bufadienolide molecules from the toad's skin secretions. The wasp can apparently also detect other volatile organic compounds released by the toad, creating a chemical signature that is specific to cane toads. This is crucial because it means the wasp can actually smell the toxins that make the toad dangerous to other predators. She is, in a sense, homing in on the poison itself. Once she has located the toad, she approaches it. Now, here's where this becomes remarkable. The toad is poisonous. Its toxins will kill most creatures that try to eat it, but the wasp does not try to eat the toad. Instead, she lands on the toad's back. She positions herself carefully, often moving around the toad's body to find the optimal injection site. This site selection is critical. The wasp must avoid the toad's own defensive glands while accessing vulnerable areas where her venom can reach the nervous system most effectively. And then she stings. The sting itself is not lethal to the toad, but what the sting does is catastrophic. The venom contains neurotoxins that specifically target the toad's nervous system. These toxins interfere with synaptic transmission in the toad's central nervous system. The mechanism is extraordinary. The venom blocks acetylcholine receptors and disrupts the transmission of neural signals between neurons. Within seconds, the toad becomes immobilized. It cannot move. It cannot escape. It cannot call for help. Its muscles are completely paralyzed, but it is still conscious, still alive, still aware of what is happening to it. This state-conscious paralysis is precisely what the wasp needs. A completely unconscious toad would spoil too quickly. A dead toad would decompose and create an unsuitable environment for developing larvae. The wasp's venom has evolved to create a precise physiological state. The toad is alive enough to maintain homeostasis, but helpless to resist what comes next. The toad lies there, unable to move, while the wasp uses the immobilized body as a food source for her offspring. The female wasp then lays her eggs on the surface of the toad's skin, or sometimes directly through a small incision into the toad's body cavity. The exact number of eggs varies, but in some cases, she may lay a dozen eggs or more on a single host. Then she leaves. She goes off to find another toad. The paralysis is not permanent. Over the course of several hours or a day, the toad will gradually begin to recover partial motor function. The toxins break down in the toad's bloodstream. Nerve transmission gradually returns. But the toad will never be quite the same. It will remain impaired. It will move slowly. It will struggle to feed. Some studies suggest that parasitized toads never fully recover even after the larvae have finished feeding and left the corpse. And inside the toad's body, the wasp larvae are developing. As the larvae grow, they begin to feed on the toad from the inside. They consume the body cavity tissue first. They avoid the vital organs initially, keeping the toad alive as long as possible, because a dead host rapidly decomposes and becomes an unsuitable environment for larval development. A living host provides steady conditions and ongoing physiological processes. The larvae have evolved what can only be described as a sophisticated feeding strategy. They consume tissue in a specific sequence. First, the fat deposits that provide immediate energy, then the muscle tissue that offers protein for growth, and finally, only when absolutely necessary, organs that are essential for the host's survival. Some larvae even produce antimicrobial substances that prevent their host from developing fatal infections that could kill the entire colony of developing wasps. The larvae are consuming the toad's fat reserves, its muscle tissue, eventually its internal organs. The toad is being eaten alive from the inside out. This process takes days, sometimes weeks. During this entire time, the toad remains partially mobile. It can move weakly, can twitch its limbs, can attempt to eat if food passes its mouth. But it cannot escape the larvae inside it. It cannot heal the injuries. It cannot mount an immune response effective enough to dislodge the parasites. Eventually, when there is nothing left to consume internally, the toad dies. By that point, the larvae have consumed perhaps 80 to 90% of the toad's body mass. A remarkable feat of resource extraction and selective feeding. The wasp larvae pupate inside or near the corpse. They undergo metamorphosis. Within a few weeks, new adult wasps emerge from the dead toad and fly away to find more toads to parasitize. What makes this adaptation so extraordinary is how specifically it targets cane toads. The wasp has evolved to handle the toad's toxins in a way that almost no other animal has demonstrated. The wasp itself appears to have some level of resistance to bufadienolides. How this resistance evolved is still an open question. It's possible that the wasps that arrived in Australia and happened to have even slightly more resistance to the toxins were more successful at preying on toads. Over generations, the population of wasps shifted toward individuals with higher and higher levels of toxin resistance. Researchers believe this shift involved changes to genes encoding detoxification enzymes, particularly cytochrome P450 enzymes, and other metabolic pathways that allow the wasp to break down and eliminate bufadienolides from its body. Each wasp that successfully preyed on a toad reproduced more offspring than wasps that couldn't handle the toxins. Those offspring inherited the genetic variations that conferred resistance. Over many generations, these variations accumulated and intensified. It's also possible that the wasps evolved the resistance more recently through rapid selection pressure, that something about the Australian environment selected for this capability within the last decade. The sheer density of cane toads may have created an unprecedented selection pressure. In Hawaii or in South America, where these wasps originated, toad populations were never this dense or this abundant. But in Australia, cane toads became a dominant prey item, sometimes the only prey item available. Wasps with better toxin resistance had access to a nearly unlimited food source. Wasps without resistance starved or died from poisoning. Evolutionary timescales are usually measured in thousands of years, but in specific ecological niches with intense selection pressure, significant adaptation can occur in decades or even years. The cane toad invasion created exactly those conditions. Intense, continuous selection pressure favoring a single trait. Whatever the mechanism, the result is clear. A wasp species that was previously not involved in controlling cane toad populations has become one of the most significant predators the toads now face. The implications of this are enormous. For nearly a century, Australia has been trying to find a way to control cane toad populations. Every method has been expensive, difficult, or only marginally effective. Direct extermination is impossible. There are simply too many toads across too large an area. Governments have invested millions of dollars in culling programs, attempting to kill toads through trapping, poisoning, and manual collection. The impact is negligible. A region might see local toad populations reduced by 30 or 40%, but within months or a year, the toads breed back to previous levels. Chemical controls are expensive and have environmental impacts of their own. Herbicides, insecticides, and toxins that kill toads also kill other animals and contaminate water sources. Some proposed chemicals are so dangerous that governments refuse to authorize their use. Biological control attempts have mostly failed. In the 1980s and 1990s, researchers tried introducing other species that they thought might prey on cane toads or compete with them, but none of these introductions resulted in significant population control. They introduced parasitic flies. They imported predatory insects. None of it worked. Some interventions had unexpected consequences. Introducing new species created new problems rather than solving the original one. The idea that a natural predator could evolve or arrive in Australia to control the cane toad was mostly dismissed by mainstream science. It seemed too optimistic. A 90-year waiting period for nature to solve the problem seemed like accepting permanent ecological damage. The cane toad seemed like a permanent part of Australia's future, a testament to human error that could never be undone. But then this wasp appeared. Or perhaps it didn't appear suddenly. Perhaps it has been present in Australia for longer than anyone realized, and its population is only now becoming noticeable as it adapts and spreads. There are reports from the 1960s and 1970s of what might have been parasitized toads in Queensland. The documentation is limited, and it's hard to know for certain, but it's possible that this predator-prey relationship has been developing in secret for decades. What is certain is that in some areas of Australia right now, cane toad populations are crashing. The wasp is spreading. It is adapting. It is specializing. This is natural selection in real time, and it is happening because of the cane toad's presence in Australia. The toad created the conditions that selected for this predator. The toad, in essence, created its own enemy. This is how ecosystems often work. They are not stable. They are not in equilibrium. They are dynamic. They change. They adapt. They respond to new pressures with new solutions, sometimes over thousands of years, sometimes over decades, sometimes over just a few years. The speed at which this adaptation is happening is genuinely remarkable. Most predator-prey relationships take thousands of years to fully evolve. The wasp and the cane toad have developed a sophisticated relationship in less than a century. But there is a complication to this story. The wasp is effective, but it is not perfect. It cannot reach all toads. In many areas, the wasp population is limited by availability of toads or by the specific habitat type. In some regions, cane toads continue to thrive and spread even as wasp populations are increasing. The toads are also evolving in response to the wasp. There is evidence that cane toad populations in areas with high wasp pressure are exhibiting different behavioral patterns. They may be changing when and where they are active. They may be hiding more. They may be altering their reproduction cycle. Evolution is a conversation between predator and prey, between attack and defense, and this conversation is still ongoing. Furthermore, the introduction of a new predator does not automatically solve the ecological damage that the toads have already done. Native species that have declined due to toad predation may not recover even if toad populations fall. Ecosystems that have been reorganized around the presence of toads may not return to their previous state. The native predators that the toads killed are gone. Their ecological roles need to be filled by other species, and that process takes time. Years, decades, or even centuries. And there is another consideration that scientists and conservationists are now grappling with. If the wasp becomes so effective that it drives cane toad populations to very low levels, what happens to the wasp? A predator's survival depends on the availability of prey. If the wasp successfully controls the cane toad and toad populations plummet, the wasp population must also plummet. The wasp could theoretically become rare itself, or even go extinct, unless it can switch to feeding on other toad species. There are other toad species in Australia. Most of them are native species that have been present in Australia for millions of years. These native toads are not toxic to the degree that cane toads are. If the wasp begins to attack native toad species, it could threaten those species as well. It could become a new invasive species problem layered on top of the cane toad problem. This is the fundamental challenge of biological control. You cannot simply introduce a predator into an ecosystem and expect it to do only what you want. Predators are opportunistic. They evolve. They adapt. They seek out new resources when their preferred prey becomes scarce. The solution to one invasive species problem can inadvertently create another. Scientists and government agencies in Australia are now carefully monitoring the wasp populations. They are tracking the genetic diversity of the wasps through DNA sequencing programs, mapping which genetic variants are becoming more common in different regions. They are studying whether the wasps are beginning to prey on native toad species through careful field surveys and laboratory feeding trials. They are trying to understand the limits of the wasp's geographic range by monitoring wasp populations in different climate zones, different soil types, different vegetation patterns. They are collecting parasitized toads, measuring how many wasp larvae develop inside them, tracking how long the parasitism process takes in different environmental conditions. Will the wasp spread to all areas where cane toads are present, or will it remain localized to cooler, wetter regions where the climate favors wasp reproduction? Will it be effective enough to significantly reduce cane toad populations across the country? Or will it only control toads in certain regions where conditions are optimal? Will the wasp's effectiveness increase over time as the population adapts further to Australian conditions? These are the questions that research is currently trying to answer through rigorous, long-term ecological monitoring. In the meantime, the wasp continues to spread. In certain areas of Queensland and northern New South Wales, the toad population continues to decline. Researchers working in these areas report finding dead and dying toads with wasp larvae inside them. They report finding parasitized toad corpses that have mummified and left behind only the wasp pupae. The ecosystem is slowly, incrementally beginning to rebalance itself. Not back to what it was before the toads arrived, because that is impossible. Too much has changed. Too many species have declined. Too much damage has been done. But towards something new. A new equilibrium that includes the wasp, the toad, the native species that can coexist with both, and the changed landscape. This is the reality of invasive species and ecological restoration. It is not a simple problem. It does not have a simple solution. It is not a story where humans intervene decisively and fix everything through clever problem solving. It is instead a story about nature finding its own solutions, slowly, imperfectly, over time. It is a story about how ecosystems respond to disruption, not with immediate restoration, but with reorganization. The cane toad is still a major invasive species in Australia. The wasp is not going to eliminate it, but the wasp is beginning to control it. The wasp is beginning to limit its spread. The wasp is beginning to prevent new areas from being completely devastated the way northern Queensland was after the toads first arrived. For anyone interested in the real story of invasive species management, or anyone living in Australia who has watched the cane toad invasion unfold over decades, this is a reminder that solutions often come not from outside intervention, but from the natural world's own adaptive capacity. The ecosystem doesn't fix itself overnight, but it does, eventually, begin to respond. The predator emerges. The balance shifts, and though the world is forever changed by what came before, new forms of stability begin to emerge from chaos.